And hello, we're back for another lab here at Hobbs High School. This last week we did a demonstration lab, and a demonstration lab is when I get up in front, excuse me, and just basically take them through what we're doing in the lab, and then they get to observe it on the overhead. In my particular classroom this year, we don't have a laboratory facility handy, so a lot of things have to be adjusted in order to get it to work. But anyway, this one is entitled Demo Lab. Now, those of you that are in class, you can look this up in your Skyward gradebook, and it's called Demo Lab. And then underneath it, we put sugars. Now, in class, we've been discussing different kinds of what we call macromolecules. And I had gone over this during class in the week, and I wanted to know if you remembered what a monosaccharide was. And so I had the students write the definition to that. And I also had them write the definition of a polysaccharide. Now, just to review, for those of you that may be watching that weren't in class, mono is science speak for one, and saccharide really, just for layman, this is some kind of sugar or sweet. So it's one sugar. So a monosaccharide is one sugar molecule. And a monosaccharide is the simplest of the sugar molecules. A polysaccharide, poly means many, so this is many, or we could say more than two or three, many sugar molecules. Why this is important, molecules, can't spell this afternoon. Why is this important? It's because different sugars do different things. For example, single sugar molecules become what we call sucrose. In this case, this is a sugar cube right here. More complex polysaccharides become the starches that we know in a potato. And here it is. I cut this one just right out of a potato. Here you go. There's the potato it came out of. Got that at my local supermarket the other night. We also, and this type of a carbohydrate is it's a little bit more complicated carbohydrate than the C6H12O6 that we learn as the product of photosynthesis. But what this becomes, this is food storage. In a potato plant, the potato creates these tubers to store energy to get through the winter, and then to push back up next year with another plant in order to go to seed and reproduce. So potatoes are what we call biennial. That takes them 2 years, bi means two, biennial takes them 2 years to go through their life cycle. So this type of carbohydrate, this kind of starch in a potato, is an energy storage device. But then we combine different forms and we can get bread. Here's the rest of the slice of bread right there. We have bread. We have crackers. Another kind of starchy plant material is called cellulose. Now, cellulose could be like the stalks of plants, the material that leaves are made out of. It's a bit more complicated than just our simple carbohydrate that we have here, polysaccharide. But even paper can be considered a polysaccharide. So we're going to use iodine. It's what we call the iodine test. And we are going to determine if we can tell the difference between a simple sugar and a more complex sugar. So I have my bottle of iodine here. Let me squeeze this out for you. The critical thing here was what color is the iodine? And all day, most students were calling that color right there, they were calling it a yellow, brown, or even a gold color. And so we're going to test these items that I just showed you. So on our paper, what we did was we wrote down potato, we wrote down paper, we have sugar, I've got a cracker right here, sugar, paper, oh, the bread, and lastly, I've got a control for this because what we're going to check is to see if the iodine, once it hits either the polysaccharide or the monosaccharide, supposedly it should change color. But I've got a control here. I also have salt. I want to know, will the iodine change colors if it hits something else that's a food that we eat? So I have some salt. There's the salt right there. All right, so here we go. Let me put the pen away here, and we'll just start. We'll bring these in to where you can see. Let me get them arranged on the camera here so that you can see some of these. There we go. All right, so let's start. So I have my handy dandy dropper here. Let's start with the potato. Let me drop some in there, and yes, it seems to be doing some kind of a color change. Let's go into the bread. Oh my, that was a definite color change right there. I have the white paper. Oh my, that changed right away. What's happening here? And then I've got the sugar cube. Well, the sugar cube didn't change color. It's just going to stay that nice golden brown on the top. Let me get out my, there's the cracker. Yep, that's changing colors. Now, just so you know, the whole fundamental purpose of this test is that when iodine comes in contact with a polysaccharide, it will change colors, and it will change it from whatever color it was, that golden yellow color, and it will change to a dark blue, brown, almost black color. And as I told the kids, I was trying to research this to find out, well, why does it do this? Well, long story short, you find out why it does this, you might be up for a Nobel Prize in chemistry, because even the scientists don't really understand why this process works. I will tell you though, remember we said this is sucrose. This is a monosaccharide. This sugar cube is several billion sugar molecules all stacked together to form that sugar cube. So when the iodine atoms were released into here, the iodine atoms just ran right by those sugar molecules and nobody interacted at all. So that's why this never changed color. But for some reason, in the polysaccharides or the cellulose, in these, the molecules, instead of being singles, they form a spiral because there's chains and chains of these monosaccharides that are all linked together. Somehow or other, when the iodine gets inside of these polysaccharides molecules, the iodine becomes trapped. Evidently, there's some type of an electron transfer, and in that electron transfer, that results in a color change. Now, that's the research that I was finding online yesterday. So if you can figure out the exact nature of this reaction, you just might be up for a Nobel Prize. All right, so my final one, I have the salt here. Now, salt, as I asked the students yesterday, is salt sugar? And students had to think about this for a second, but usually in every class there was one student that knew that salt was sodium chloride. Remember, in these, the chemical formula is C6H12O6. That is the general chemical formula for a carbohydrate. But in salt, this is NaCl. NaCl is its own different molecule, sodium and chloride. This is carbon, hydrogen, and oxygen. So salt, of course, is not sugar. But I'm going to use this as a control. I want to know, will this react with the iodine? This is a food. It looks white, but no. As a matter of fact, most kids says, well, that looks like an egg, Mr. B. And so sure enough, it does. So as we look down on here, there's our results of our experiment. Of course, here's the sugar cube over here. The sugar cube and the salt did not change colors, whereas everything else, these four items, the potato, the bread, the cracker, and the paper did change colors. Going back to the paper for a moment, students wanted to know, well, if paper is a polysaccharide, why doesn't it taste sweet, or why doesn't it have a taste? Paper is made in a process usually using trees or other pulp paper products. It's put in a vat, slightly acidic solution, and it's turned into a pulp, about the consistency of a really watery oatmeal or cream of wheat. Then it's run through the process where the water is taken off, it's heated up, put under a little bit of pressure, and the cellulose molecules are forced back together and recombined, and then the final product is bleached, and that becomes the paper that you have. So on the paper, we put down here that the potato turned black, the paper turned black, BLK, the sugar turned black, the sugar did not change, there was no change, sorry about that. The cracker turned black, the bread turned black, and the salt, there was no change. So, final question was, were we able to find, indicate polysaccharides? And the answer to that was yes, we were. Then I asked the students in a paragraph, what did you learn today? Now in my classroom, one paragraph is equal to 5 to 7 sentences. And everything from here up, this is 50% of the lab grade, this is the other 50% of the lab grade. And I will deduct 10 points per sentence that is missing. And I had a couple of students say, well, our English teacher said a paragraph can be as little as 1 or 2 sentences as long as it gets your point across. And I understand that, and I taught English for many years. But in my classroom, I want to know if you can explain why you think the way you do. And that should be able to give me a topic sentence, 3 reasons why, or 3 examples, and then you should be able to give me a conclusion. There's your 5 sentences right there. It's not that difficult, but this was the lab that we did in class on Friday, October the 5th, 2018.